I believe, Logan, you're up first. Just remember, Nick is presenting again, so if anyone wants a return compliment question, now's your time to start thinking.
Cheers, thank you. Perfect. I might have got a bit longer, sorry. Hey guys, so my name's Logan Hellmrich. I'm a PhD student for the Deep Reefs here at the Ningaloo Outlook 2. I'm doing my PhD with Curtin Uni through the Fish Ecology Lab. So I'm going to do a brief sort of run through on all my chapters, so not going to be too much on each of the results. But yeah, quickly, so although with the Deep Reefs, we're focusing on mesophotic habitats. So these are habitats that are low light and intermediate depths, so normally occurring at 30 metres down to the lower photic zone. So these habitats globally are understudied. However, they're quite important, and in particular at Ningaloo, because more than 50% of the Ningaloo Marine Park consists of depths greater than 30 metres. So it's quite an essential area that we need to know about. The last Deep Reefs team looked into the habitats and looking at mapping them and describing the distributions of them. So now that we know where these habitats are, we're able to go to these habitats and look what's happening at them and take a deeper look at the ecology. Cool, so we're able to pick out 6 distinct habitats from the previously described habitats from the last Ningaloo Outlook. So the first one is coral. So coral's pretty obvious. We surveyed this from 18 metres up to 35 metres. So we know coral's quite abundant below 20 metres, but it is still quite evident around these deeper depths. Our next site was sponge. So this is where the coral starts to decrease with the low light. We start seeing these sponge gardens and these filter feeders sort of take over on these softer substrates where corals don't tend to be found. We surveyed these from about 35 metres up to 50 metres. Next habitat is sand. So sand we surveyed from 35 metres up to 70 metres deep. It's probably one of the most important habitats due to the fact that it's the most abundant one out the back of the reef, and in particular in between all the habitats. Although in saying that, trying to find just a boring section of sand to survey was quite hard. We'd always land on a bit of coral or something else, so that was the trouble child, this one. Our next one was macroalgae. So we had macroalgae from about 20 metres up to 40 metres. So in the more shallower sites, we had these green kelp-like macroalgae. And as we moved deeper, we had the more red, fleshy macroalgae that was in between the rubble and the Cyclocereus, which is another habitat. So the rhodolith is another habitat that we surveyed, which ranged from 25 metres up to 40 metres. This is a free-living coralline algae, which the video just crash, but it's just these purple rocks pretty much. And the last habitat is Cyclocereus, which is a mushroom coral. So these are solitary mushroom corals, which only occur in the northern end of Ningaloo near Helby Bank for about a 2 kilometre stretch. So you can see this is just a full dense field of the Cyclocereus, which is they're solitary mushroom corals, so they can flip up and move around, but John will go into them in a bit more depth. So to survey some of these habitats, we used ROVs, so remotely operated vehicles. So pretty much they're an underwater drone. So the SRV8 is one that we have, and we control it with a joystick, so it is tethered back to the boat the whole time, which feeds us a live stream of where it is at. Well, the camera, and then we can also have the positioning system, which shows us where it is in relation to the boat. So it's quite a new technology, but it's giving us the ability to almost pull divers out of the water and put ROVs in. So ROVs can go to depths greater than divers. They can have much longer dives, and we can use an ROV all day. So they're quite cost effective. We don't need a dive team. We only need a minimum of 3 people to operate an ROV, and there's no diving costs and stuff like that. So it's the one outlay for the ROV to begin with, but overall they're quite cost effective, and it gets over some of the logistical constraints of diving, and in particular in my work, the deep stuff where we can't really dive, so we send out an ROV that we can just explore around in. Then we add on these stereo cameras, so same as what Anthony was saying, so we add these onto the ROV. So this allows us to conduct transects, so we can get the 3D measurements and lengths of the fish and other animals. So we're able to get a paper published, which was my first paper, of pretty much an ROV methods paper, and it compared it to doves, stereo doves in particular. So looking at the results really quickly, we pretty much found that the two techniques found statistically similar results and highlighted similar multivariate patterns between sanctuary zones and lake location. However, we did see a major decrease in wariness towards fish, towards the ROV. So fish tended to be less wary towards the ROV, getting closer to the ROV and fleeing less. So we saw less flight responses towards the ROV. So we were able to put this together to say that these two methods can complement each other. So we can continue data sets that would normally have divers going. We can now use ROVs without messing up the data set. However, we do have an obvious decrease in a bias towards divers, so the ROV helps eliminate that. So we're able to use the ROV to identify habitat users at these 6 habitats that I just explained earlier. So those videos were actually the ROV conducting the transects. Most of our sites are around the northern Helby Bank area simply because that's where the habitat is, and then we're able to get some of these sand and coral sites as we move down. So this here is just pretty much a typical ROV transect. The ROV hovers about half a metre off the ground and slightly angled down so we can film the fish or anything that's really down there. So the fact that it is a transect-based method, we can actually make fine-scale habitat associations with this. So we can go, these fish are with this habitat. We're not attracting them in. We can link it exactly with this habitat and look at the fine-scale changes. We can also use the ROV for other methods, not only just counting fish. So we're able to hook a fishing net on and hook up one of these short-spined crown of thorns. So John, I won't go into this too much depth because John will talk all about that. We've also been able to put a grapple hook onto the ROV. Looks pretty mean like that. So this goes down, it's able to grapple onto objects, pull off, so then the cord is free, and then we can winch it up from the boat, because the ROV does have a hand on it which can pick stuff up and move it, but stuff like these bricks are quite heavy, so the ROV doesn't have the power to pick it up. So those things down is one of my next chapters, which are these settlement collectors. So we had them out again at these 6 habitats. So the 6 habitats that we have, we keep going back to each time and use our different methods to have a look what's happening there. So as you can see, it's a brick with the 2 terracotta tiles, which are to promote benthic growth, so we can see what's happening there. And they have these cages, which are full of these plastic biobowls, which are to promote the recruitment of more invertebrates and organisms. And then simply the float and the rope is just to float up so the ROV can hook into it. So this will help us find what are the key habitat forming species that are coming into these habitats. So as you can see here, these are some of the tiles that we got photos of. So this is what we'll be analysing with dots, random dots, which we will then identify what is there. So it's pretty obvious with some of these sites. We have a sponge site, we have sponges growing, macroalgae site, we have macroalgae. At a coral site, we have soft corals. And at rhodolith, we have these purple rhodoliths. In terms of the biobowl cages, Wilmore, so this is how they started and that's how they ended up. So for over 7 months, there was quite a lot of growth. We weren't too interested in what was happening on the outside, but it was still interesting with what was growing on there. It was more what was inside and what communities had recruited in. So we had, in such a short period of time, so many animals and little invertebrates that came in, from crabs to little prawns, to sea cucumbers to octopuses to heaps of juvenile fish. So it gives us a look into what is coming into these habitats. So we're able to process these on the boat. So you can see that's how much gunk was pretty much inside the cages. And then we process them in the lab. So we're able to take a sample of filtered water for eDNA work. So thanks to Cindy for helping with that. And then we washed every single bowl. So I managed to wash every single bowl. So it was almost, there was 2,700 bowls that we washed individually to get all the gunk out, the little crabs, the little fish out of them, which we were able to pick out most of the organisms, and then we sieved them through. So the top 2 tended to have most of the big organisms, and then the rest fell through. So we've got all them stored now, so we'll go through them and look at them under a microscope and then identify what's there. For one of my next chapters, we did a conventional BRUVS trip, so baited remotely underwater videos. So it's just a typical monitoring tool. However, in comparison to ROV, now we're stationary and we have added bait. So we're attracting more of these predatory, more shy species in that we don't normally see with the ROV. So have a look at here. This is just a typical BRUV video. So we've got heaps of fish coming around. It's just chaos. But this is part of the reason why we've done it. So we have this site like a sand habitat. We normally run a transect across there with the ROV. We wouldn't see any reds or any targeted species. And straight away with a bit of bait, we're attracting in these targeted species which are in the area. Another example of that here is the Cyclocereus beds. Again, with the transects, we've probably only seen about one targeted species. Straight away, we got 2 species in the shot. But in saying that, we do also get the more predatory species like sharks in, which do sometimes take the whole bait bag. But we do actually get them in the study now too. So we do see them with the ROV, but they tend to stay at quite a distance. It makes it hard to identify. And then we have hungry fellas like this guy that comes in, so sort of pushing the idea that we're getting the predators in again. He doesn't mess around. So we thought we were dragging the bruvs up and ruining them. Turns out they were just taking them. And honourable mention to the Northwest Blowies, who pretty much ruined every single bait bag that we had by the end of the trip. So then finally, we did these long runs. So long runs that we were just talking about with the last talk. So these are long run remotely operated videos. So pretty much the exact same as a bruv, but we're just taking the bait off. So these cameras run for a full 24-hour cycle, so we can see the influence that the day-night cycle has on habitat users again. So we know that the diurnal cycle, so as we move from day to night, is one of the biggest influences of change in the marine habitat. So we thought doing this would just take us the next steps. So instead of doing a snapshot of what's happening during day, we can look at what's happening all up. So just a quick comparison, we've got a bruv here on the right and then the long runs or ruvs. So we pretty much just pulled off the bait pole, put some bait on there, and then we've put on another housing which has a pretty big battery, which allows the light just to stay on for 24 hours too. So light stays on and the cameras record for a full 24 hours, which will come back and take out the clips. The plan is at the moment to analyse 10 minutes every hour, so we can have that full 24-hour cycle. And this here is pretty much why we've done it. So we see this sponge site, not much is really happening early in the night. Move out to a couple hours later, we have these predatory fish coming out now. Move on a couple hours, they're still hanging around, but we've got different ones now coming in. And then we'll move into the early hours, and we have a whole different assemblage of fish using this habitat. So if we were to run an ROV transect across here, we would probably just see these fish and assume that these fish are associated with these habitats. In reality, there's a lot more happening. And I had to include this one because the Spango's pretty cool and just looks like he's talking to the camera. Showing off. And he stays there for a couple minutes. I can keep fast forwarding and he's still there. But yeah, we also get habitats like these rhodoliths. See if this one loads. So again, if we drove an ROV across here, we don't see much structure. We wouldn't actually expect much to be happening here. We rarely see any fish like this with the ROV, but early hours of the morning, we're seeing all these juvenile emperors come in, highlighting that it is quite an important habitat, watching them actually feed so we can have a look at their behavior. And then one last comparison. So this is the sand site. Not much is happening during the day. We have these little eels coming up, which is pretty cool. Move into the night. We've got some footage of some squid coming in. So these squid are actually laying eggs. So we can see them actually headbutting the sand. So they hang around for a while and pretty much cover the floor in squid eggs. And you can see how many squid eggs are there now. And then a couple hours later, we see the spangos come in and they're eating the squid eggs. So we're not just seeing fish, we're seeing their behavior. We're seeing like the whole story come together now. So we're linking this together with the ROV transects and the bait, bringing in the predatory fish and stuff like this. We're starting to get a clear image of what fish or what animals are pretty much using this habitat. And it's saying that it's not just fish, we're seeing all different types of animals too. So the fact they're on for so long, we can get a full picture. So we have a turtle down here at this almost 40-meter site. It comes down to say hi again. And then he takes off. And then lastly, there's this little mantis shrimp we've got on a time lapse. You can see him moving all these rocks around, making his little home. And for hours, he goes out, comes back, goes out. So again, just highlighting, we're not just looking at fish. There's so much other stuff happening that we can observe with these 24-hour cameras. So that's it. So thank you. I'd like to also thank my supervisors, John Keesing, Ewan Harvey, and Ben Saunders, as well as everyone that's helped me with the field work. So you guys all know but Nick, John, Cindy, Bo, Ryan, Damo. Thanks for your help. couldn't have done it without you. Thanks, guys.
Good afternoon, everyone. It's great to see such a great turnout for this symposium. So thanks everyone for coming along. I'm just going to talk to you briefly about some of the things we've observed on these mushroom coral beds that Logan talked about before. They form very extensive habitats in that northern part of Ningaloo at about 40 metres depth. And in particular, I want to talk about predation on these corals by short-spine crown-of-thorn starfish. So there are two types of crown-of-thorn starfish. Well, there are actually about five species, but there are two main types. Pacific crown-of-thorn starfish, which is the one that's notorious for cycles of coral mortality on the Great Barrier Reef and elsewhere in the Indo-Pacific. And they occur in high densities up in the Pilbara, and we've done some work on that in the past. They are rare at Ningaloo. They've, as far as I've been able to tell, they are present there, but rarely observed and over a long period of time. The short spine crown-of-thorn starfish, probably not as well known, Acanthasta brevispinus. The first record for this species was at Ningaloo. It was in 2006. A team from AIMS and WA Museum collected just a few individuals, and this is a bit of a sort of contrast of the biology of the two species. Short spine crown-of-thorn starfish generally regarded as being deep water, soft sediment habitats, predating on things like scallops, carrion scavengers, not known to outbreak, only known from at low densities, and until recently hadn't been observed sympatrically with Acanthasta planki. So this is sort of the traditional view, going back to probably up to about 5 years ago, of how these two species shared or didn't share a particular habitat, but partitioned habitats on coral reefs and made use of the resources on coral reefs. Okay, so in 2013, AIMS team led by Andrew Hayward on using tow video found there's actually 6 of these in this picture, so we don't know how many there were, but that's a reasonably dense aggregation of the short spine crown-of-thorn starfish, and they seem to be associated with these mushroom corals, these Cycloceras distorta, but wasn't possible from them to tell whether they were eating those corals or not. So this was in 70 metres of water on Rankin Bank. As part of Ningaloo Outlook, we were actually able to demonstrate that these short spine crown-of-thorn starfish are consuming the mushroom corals, and this was actually the first record of the species eating hard corals, and you can see on the left there, the mushroom coral actually in the mouth. The way these things feed is they avert their stomach over coral. So when we've scooped it up with the ROV, it's contracted its stomach, but it's still got the coral in its what makes for a mouth on a starfish. Just note the short arms up that top right-hand picture is an indication that something is actually biting off the arms of these short spine crown-of-thorn starfish. We don't know what that is, but it's probably some sort of fish. So around about the same time, Dan Godoy found that Acanthasta brevispinus were also consuming solitary corals on the southern part of the Great Barrier Reef. These small heterosamnia. The red arrows in the picture show where the those corals are actually quite dense in this particular habitat, and he found they're actually quite about 8 per 100 metre square. It was actually quite dense, but it was very limited, still a very limited population of only several hundred individuals. A few years ago, in southern Japan, the starfish was found for the first time to eat soft corals, in particular Dendronephthalia. And this was in a habitat where there was plenty of hard coral. The other interesting thing about this study was that it's the first to show that Acanthasta brevispinus and Acanthasta, the Pacific crown thorn starfish, were sympatric, or occurring in the same habitat, and these including at depths less than 20 metres. So there've recently been a bunch of observations on the feeding of Acanthasta brevispinus, including ours at Ningaloo, which have sort of changed our thinking about the comparative biology of these two species. That they're probably not so different after all. They certainly occur sympatrically. We found them at reasonably high densities. They will eat soft corals, they will eat hard corals, and they're certainly not restricted to soft sediment habitats. And the short spine one's going to occur in quite shallow water. We haven't seen them in shallow water at Ningaloo. Our observations have been limited to the Cycloceras beds. This is just showing how extensive these Cycloceras beds are. So these are very small mushroom corals, only about the size of a 20-cent piece. But it seems to be not only extensive habitat, but as Logan's just shown before, a quite well utilised habitat. So mushroom coral beds at Ningaloo. I just said they form large, often monospecific beds, not just at Ningaloo, but elsewhere, not just this species, but others. And we're looking at the importance of these habitats at Ningaloo as part of Ningaloo Outlook. And it's still not clear the extent to which predation by Acanthasta brevispinus is important or just interesting. Just want to thank my co-authors on this paper, and all the people that helped with the field work that Logan mentioned before. Thanks.
Two times. Right. There we go. I actually gave up drinking about 10 years ago. I was in training for a pole dancing competition. I got best costume. It's the only athletic award I've ever had. Well, if you can call it an athletic award. Let's think. I did get most improved cheerleader one year. Yeah. Here we go. Okay. So, I'm going to start this watch. So, yes, long-run cameras. Logan mentioned long-run cameras. A while ago, as PIs always do, John came to me and said, I want to have some long-run cameras. I want them to go for 24 hours. And we had a few ideas, and then it was long-run stereo cameras, and it went on from there. So we settled on some GoPros, and we've actually got 16 frames. That's 32 GoPros. Logan showed you this picture here. We've got a torch in the middle. That's just on all the time. It'll last about 24 hours. And we're going to deploy these, and we thought, what is a good method of deploying these if you've got 32 cameras to marshal the data and keep things going? When luckily we found that GoPro introduced these new QR codes to allow you to set up the cameras. So these QR codes, if you download the GoPro firmware and update it. Allowed you things like set the time, so I can use my mobile phone and hold this pattern in front of the GoPro, and it'll set the time on the GoPro. When you've got 32 cameras, you can just march around going like this in front of all 32 of them, rather than like, oh, Bluetooth connect, set time. Yeah, so it really has made things more efficient. And also they have this GoPro QR code coordinator, and you can set just about anything, any function you want on that GoPro and set it into the QR code. Things like whether I want what frame rate I want, what resolution. Notice I always select 1080p, because if you pick a higher resolution, it always overheats and stops. So, but ironically, 4K of fish a meter out from the camera isn't really that better than 1080p. So we work on 1080p. Once we get all these things, I'll just go down to the bottom. And what is really cool is this box here, additional commands, allows you to type in other commands you'd like it to do. And my favorite command, wait for it. My favorite command. Commands always start with an exclamation mark and S. Start recording. So whenever you show it that QR code, it will start recording in the mode that you want. And to prove it, we've got the subject, Logan, caught on camera showing a QR code to a GoPro. Yes, and so when we've got all these cameras in the thing, next becomes the problem, and this is where I come in as a sort of software-y, engineering person. This is the end of the day's labor. 32 SD cards, all of them 512 gigabyte long. And when you do the calculations, it suddenly came to me that it would actually take us to download all these on the standard network connection of 100 meg. It would actually take us nearly 2 days to download 1 day of vision. And I'm going, at this point, I go to the internet and find what is the fastest hard drive I can buy that I can take with me. Now, believe it or not, this does actually fit in as carry-on luggage. But just don't let anybody pick it up, because they go, oh no, that's overweight. But we've got basically 18 terabyte drives in there, striped. And then I went and got the fastest SD card readers I could find. And these SD card readers basically they give about 5 gigabits per reader out. And then you have to work out that you want to read one card from each pair and then keep going. Because, I mean, most people like me, I like to sleep when I'm doing field work. And I meet all these sleep-deprived students of brubs that go like, oh no, in my day, we just had to stay up all night downloading the vision. It's like, no, I like to sleep. So if you like to sleep, you have to learn to write more code. Because write some code, and then we're going to field test it. I'm going to do a field test with this code, and look at this. This is one of the most beautiful things. It's worked out where the cards are. I've got 16 cards loaded, and it's going to pick which cards and which pairs of readers they're in, and then I'm getting a full 800 megabits per second coming out of those card readers. And I'm thinking, like, that's only going to take a couple of hours to download those 16. So by the time I'm ready for bed, I can plug the next 16 in, hit go, and go to bed. And the next thing that is really important is you automatically wipe those. Have you ever come back from a field trip with like cards with stuff on, you find them 2 years later, and you're kind of paralyzed. You think, I can't delete that card. I can't. Yeah. So, having everything cleaned by computer before you start. Did I say write more code? Yeah, that was it, wasn't it? Write more code. So you need to write more code, because what you want to do is take in this kind of like, I've got 32 cameras, and everybody says, oh, I should mark. I'm sort of a, I don't know, my daughter reckons I've got ADHD, but I'm a dyslexic or whatever it is. But anyhow, the thought of mentally tracking 32 cards and working out what camera they came from is just like mind blown. So what I have is a spreadsheet here, and it has all the camera serial numbers. And I go and get the serial numbers from the EXIF data from the video, and then write some more code. And lo and behold, it gets me all the cameras and stores them in the right directories with the camera number, the serial, and the start date. And I can have a look at that before I go to sleep and think, have I got a problem? Well, probably not. So next, I write more code. because that's still not good enough yet. So I'm going to write more code because often the time goes wrong. So now I've got a little spreadsheet. I can just enter the time offset. So if I've done something wrong and that's the wrong date, even though I've got the camera setting, somebody's done something, or even the GoPro's just got it wrong. I had one GoPro that said it was the same day for 4 days in a row. Well, I don't know. His clock just stopped. So then I say, okay, to the biologist, where did we go? Fill in this form here. And once they fill that in and I run some more code, all of a sudden, all those files turn up in the right place with the bars all labeled and nicely addressed. And at the end of the week, we had over 9,000 videos. And the 9,000 videos is kind of fun because with the QR codes, you can set it into long chapter mode. And in long chapter mode, you get 12 gigabytes per file instead of 3. So you got effectively 4 files in one, which is really nice. And that gets around the other thing. The GoPro only records 100 files. That's why it's called GoPro 100 in directory. It only records 100 files per sampling. So you can actually get 24 hours if you use the QR code. And then we had over 100 terabytes of video. And it's ready for analysis. And we've got at least 4 students working on this. And credit to Cindy and everybody and to Logan. We've sort of, and Erin, is Erin here? There's Erin. Erin has basically been looking through this. Erin's from the UK. We gave her a month. Here for 3 months, 4 months, 5 months? 4 months. 4 months and counting. And we've got urchin movies for that. So we have promised her some field work, though. So you will get out of the office, because I think that's what's really important about these things. Me as a computer science sort of data guy, actually getting out into the field and actually making this work. I see so many times where people write scripts or say, oh yeah, they design something, give it to you, and you go into the field and it just doesn't work. And what's worse than it not working in the field is when you come back and say it didn't work, they don't believe you. So a quick shout out to the team. I think that's most of us there. There's always a few people who cycle in and out, but it's a really good team, and we've had a lot of fun doing this work. 8 minutes.